Yay, we are to the last lecture for chapter 10. And in this lecture, we're going to be comparing mitosis and meiosis. So remember, for animal cells in particular, mitosis is the division of somatic cells. And remember, somatic cells are body cells. So mitosis is essentially starting with a parent cell. Most of the time, that cell is diploid. There are some organisms, you'll learn more about them in Bio 2, that may have haploid cells that divide by mitosis, but most of the time we're talking about a somatic diploid cell, whether that is animals, plants, fungi, protists, anything. And so the goal of mitosis is to create two daughter cells that are genetically identical to the parent. Okay? Now remember, prior to the start of mitosis, cells have been in interphase, and during S phase and interphase, they replicate the DNA. So in these examples in this figure right here, we are starting with a diploid parent cell with a diploid number of 6. Okay, so if we look at this parent cell prior to DNA replication and then after DNA replication, we see that we still have a diploid number of 6, again, because we have 6 centromeres. And we count chromosomes by counting centromeres. Each chromosome after DNA replication is a pair of sister chromatids. Okay, so in prophase of mitosis, the chromosomes begin attaching to the mitotic spindle. In metaphase of mitosis, the chromosomes are lined up single file on the metaphase plate. And then at the end of mitosis, we have 2 daughter cells that are genetically identical to the parent. Okay, well, in meiosis, on the other hand, we are starting, in this example, we're starting with a diploid parent cell. Meiosis always has to start with a diploid parent cell. Okay, we have DNA replication during S phase of interphase, and then in prophase 1, remember we've got those 2 unique things that happen. We've got synapsis, where the homologous pairs of chromosomes find themselves, they pair up. And then we've got crossing over, where the non-sister chromatids physically swap portions of their DNA. Right? Another important thing that happens during prophase 1 is the meiotic spindle starts forming. And remember that for meiosis, the meiotic spindle fibers are attaching to each chromosome in the homologous pair, not to each sister chromatid like they did in metaphase of mitosis. So look here, we've got in metaphase 1, when the homologous pairs are lined up on the metaphase plate, each chromosome in the homologous pair is attached to the spindle. Okay, because those sister chromatids are sharing a common centromere. Now remember how these homologous pairs of chromosomes line up on the metaphase plate is completely random. All right, we've got that random alignment. Then in anaphase 1 and telophase 1 of meiosis 1, the homologous pairs of chromosomes are separated. They're pulled to the poles of the cell. The nuclei begin to reform, and then cytokinesis typically happens. So at the end of meiosis 1, we have haploid cells, and we have 2 of them, 2 haploid daughter cells. But each chromosome in each daughter cell is still a pair of sister chromatids. Well, then Each haploid cell enters meiosis 2, which is subdivided into prophase 2, metaphase 2, anaphase 2, telophase 2, and cytokinesis. The goal of meiosis 2 is to separate the chromatids. Okay, and that happens during anaphase 2. Anaphase 2, because it is separating the chromatids and getting them ready to be in their own individual daughter cells, anaphase 2 is where independent assortment happens. Independent assortment. Okay. And then at the end of meiosis, we have 4 haploid daughter cells that are not genetically identical to the parent. Okay, so meiosis. How are 4 haploid daughter cells that are not genetically identical to the parent? Let's talk about how they are not genetically identical to the parent. Number 1, they are haploid rather than diploid. The parent cell for meiosis is always diploid, okay? The daughter cells are haploid. That's one obvious way that the daughter cells are different. Another way that the daughter cells are different results from crossing over. And again, that happens in prophase 1. Okay, another reason that the daughter cells are different, genetically different from the parent cell, is a result of independent assortment. That happens in anaphase 2. Okay, so those 4 haploid daughter cells are not genetically identical to the parent cell for these 3 reasons. Okay, now crossing over and independent assortment increase genetic variability. We've talked about this a lot. Another thing that increases genetic variability in populations that reproduce sexually is random fertilization. Now, I said earlier that the parent cell in meiosis always has to be diploid. Well, why is that? Remember a few lectures ago, I'm not sure how many, 2 or 3, I told you that another name for meiosis was reduction division. Meiosis is reducing the chromosome number from a diploid parent cell to a haploid daughter cell. Well, you can't take haploid cells and reduce the chromosome number any further and still have functional viable cells. Okay, so meiosis has to start with a diploid parent cell. Now, the diploid parent cell in meiosis is a germline cell. Okay, so it's only the genetic information that is found in the germline cells that are passed on to future generations. So, when we talk about maybe diseases running in families or genetic traits that run in families, the genetic information for those traits or diseases has to be found in the DNA that is in the germline cells of the individuals in that family. Remember our example about, melanoma resulting from too much exposure to the sun. Those mutations as a result of sun exposure are happening in somatic cells. They are not part of the germline, so they are not passed on. But if we're thinking about cancer as an example, there are some genes, in particular, we know of some genes that are associated with breast cancer that can be passed on in families. And so, for example, when my mother was diagnosed with breast cancer, she underwent some genetic testing to see if her cancer was caused from a couple of different genes that are known to be heritable, that run in families. One of them was the HER2 gene, and the other one was the BRCA gene. Because if she was shown to have those, the versions of those genes that are implicated in breast cancer, then I would need to be tested to determine if I had the versions of those genes, the alleles that are implicated in breast cancer. It turns out that she didn't have either one of those. And so as a result, my risk of developing breast cancer is not necessarily higher just because my mom developed breast cancer, because the genes that we at least know of right now that are heritable for causing breast cancer, she was negative for those alleles. Okay, so basically what I'm saying is that whatever genetic abnormalities led to my mother's breast cancer, based on what we know right now, those alleles were not part of her germline. And as a result, I did not inherit those. So it's just important to distinguish between germline cells undergoing meiosis and somatic cells